Good, Randal, can you switch your webcam on?
My webcam is on. If you can't see
good,
it,
here you are.
It's... Okay, I would like to hide that window though, because it allows me to see myself. Okay.
Right. Oh, you have put yourself on a screen and I put on another, so now you are in two screens. Okay, let's leave it like that.
I'm on two screens? Oh, I don't remember putting
why
myself
not?
Anyway.
Some advice to others. It's always good to keep the microphone switched off when the presenter is speaking. So when you want to ask a question, we switch the microphone on. And if you have a webcam, you may want to switch also the webcam on and put yourself on one of the two screens, like I'm doing now. I'm opening the video feeds window in the to see all the people with a video feed. And I put myself on the screen on the left. That's a nice thing to do. That you can do when you want to ask a question. We switch the microphone on and you switch the webcam on if you have one. I will remember to put you on a screen if I see you on webcam. I have started recording already, so Randal, you have the floor.
Thank you. Okay, so what I'm going to talk about today is more of an overview of everything that we've been discussing for years. So I'd like to introduce some new terminology, at least for those of us who haven't been discussing this recently, and also I'd like to introduce some new organizations, and I would like to talk about the challenges ahead. For that reason, I'm going to go over a lot of known ground, at least for most of us here. And nevertheless, if any of you have any questions, if you're new to this topic, please just shout out. I don't mind stopping the talk and answering questions and then continuing. I have no problem with that whatsoever. So I've given this talk the title Realistic Routes to Substrate Independent Minds, which is also the tagline of a new organization called Carbon Copies. And I would like to start by explaining what that actually means, why we call this Substrate Independent Minds. And let's see if I can page forward. Okay, so this is just a quick overview of where we're going with this talk. I'm not sure how much of this we'll actually have time for. We may have to hurry up in the end because there are quite a few slides here, but I did want to kind of split it into three parts. I wanted to talk about what it actually means. What does substrate independence mean? What are we trying to talk about here? There are other terms that are associated with that, like whole brain emulation, and I'd like to make it clear what we're actually discussing. Then there's the question of why. So why do you do something like this, or why do you strive to achieve something like this? And how. So this is very important, and it's also the longest section, is how do you actually go about it? How do you get something done? That's an important topic for our group because this is meant specifically to be an action-oriented group. This is not a discussion group, not a philosophical group, but a group of people trying to achieve something. Okay, so that's the first question then. What do we mean by substrate independence? Why do we even have this new name for what used to be called whole brain emulation and perhaps colloquially mind uploading, though that's really not a very useful term. Well, the problem is, first of all, okay, just to talk about the term for a second, whole brain emulation is a very nice technical term for an approach, an engineering approach, I guess I would call it, to what I will discuss in just one moment, but we needed something to describe what the actual objective is. So not a specific approach, not a specific method, but what is it that we strive to achieve? Substrate independence. Well, let's start with the Church-Turing thesis. The Church-Turing thesis, basically, if we boil it down to a sentence, it states that everything computable is computable by a Turing machine. This is so well known now. It's universally accepted pretty much. I mean, there are always a few outliers, but pretty much everyone accepts this. Similarly, in neuroscience, there is also a consensus. And the consensus that exists, quite a rational one, is that the mind emerges from classical biophysical processes of the brain. This does not mean that there can't be any quantum effects anywhere in the matter that is the brain or in the physiological processes if you dig down deep enough. In fact, you would find that there are effects such as that throughout the universe. But what it means is that we don't need to rely on anything exotic to bring about what we consider the mind. That in fact, all of the process that we can observe, all the behavior, the characteristic behavior, can emerge from the simple responses of these cells that make up our brain. So, if these processes are computable, then that means that the mind is computable. In other words, our brains are machines. They're complex, but they are nevertheless machines. Now, the Church-Turing thesis implies that one Turing machine could implement another. We can think, for example, of emulating a PC on a Macintosh. That's an example that's pretty understood now. Similarly, therefore, it should be possible to do that with the human mind. And this is what we mean by substrate independence. You can take something that is running what we call the mind. Right now, it's our biological implementation in the brain. And we could move it to another platform. We could be able to run those same functions on other hardware. Oh, I just shot past something here. Okay, so let's go back to that term whole brain emulation. This was something that I cooked up a number of years ago, and it was meant to describe a process. It's how do you go about putting the mind in a different substrate. So it's not the fundamental idea of what we're trying to achieve, but it does talk about one way of achieving substrate independence for the mind. Enabling the transition of the functioning mind from biophysical circuitry to other computational circuitry is the objective of achieving substrate independent minds. Now, the most conservative target is whole brain emulation. Why is this the most conservative one? It's the most conservative one because it attempts to mechanistically re-implement all that which enables predictions of active state and behavior at one time, at a time t plus delta t, so a little bit later, from what you know about the behavior and the activity at time t, with an acceptable error. And I won't talk about the error today, but we'll have to define at some point what is acceptable. So it's a very conservative approach. It just says we know that it works on this hardware, so why don't we try to re-implement all the basic functions of that hardware? Okay. Now, what is Carbon Copies? Carbon Copies is a new organization that is going to support research as well as development towards or advancing substrate independent minds. One of the things that we intend to do is to develop a good base of literature that examines the fundamentals, because I believe we need to take a little step back. This doesn't mean we can't continue to connect with a lot of projects that would like to move ahead, developing specific procedures that are useful for developing substrate independent minds. But what I do think is that we need to take a look at what are the underlying fundamentals of the objective that we're talking about. What exactly does it mean to have functionality of a mind? What does it mean if we want to move that to somewhere else? What needs to be preserved? There are all sorts of specific questions, some of which I'll get into. We also want to be able to produce peer review, that is, induce or elicit peer review through those publications and through events. The thing is, we're really interested in the objective. We're not trying to push any ego or anything of that matter. We really want everyone to point out all the possible mistakes we might be making. Any comment and any error, we'd love to know about it, because everything like that can help us forward. And also, we want to develop and track a roadmap. So a living roadmap, really, and an approach that tells us, okay, this is working, that's not working, where do we have to go, what's the next milestone, and what kind of resources are needed in order to get from A to B. So to summarize what Carbon Copies does, really, Carbon Copies cares. Carbon Copies cares for projects. In other words, we care for the people who want to work towards substrate independent minds. We want to care about what are feasible and validatable methods for subject-specific acquisition of data at large scale and high resolution. That's just one example of a project that might be interesting. or various projects of a commercial nature that would, for instance, do something in cognitive augmentation, and a number of others you can imagine. There are so many possible projects, some of which already exist and which should be somehow linked to efforts towards substrate independent minds, and some of which need to be developed, new key projects that should appear. Also, we care for the larger community, meaning we care not just about what's going on in these projects, but we care about what comes out of them. We care that there is an honest and rational evaluation of results. We care that there is a clear and open explanation of our objectives, and that we also reach out towards the larger community and listen to their comments and their concerns. So this is a dialogue. We have a network, we foster this network, we go out there, we present ourselves, and we talk to people. This is a very important aspect of what Carbon Copies is about. Okay, now let's look a little bit at the why. There are a number of things to consider there, and the first I want to do is take a little look back into history, the short history that there is right now, of the origins of Advancing Substrate Independent Minds. One of those origins is scientific. It's a desire to understand how the mind emerges from the functions of the brain, and to explore and discover things such as the human connectome. This is a very hot topic right now. To understand the effects of structure and function in the mind, which of course, as in all neural networks, are closely entwined, and therefore it's a highly interesting but very novel thing to understand compared to, say, traditional computing structures. And we want to understand all of this in a modular brain system. We know that the brain is modular, and at a large enough scale so that we can incorporate many of those modular regions, and at a high enough resolution to understand what's going on locally within each of those modules in the large networks of neurons that are necessary to drive function. So all of these are topics that are really interesting to neuroscience at large, to psychology at large, cognitive science at large, and many others, including AI. These are not new areas of interest, but these are overlapping areas of interest, overlapping disciplines with that which we're trying to do. So maybe it's useful to take a really quick little detour and give a brief overview of the brain for any of the people out there who maybe perhaps aren't as familiar with these underlying facts, and maybe don't come from a neuroscience background or something related to that. So the first thing I'd like to say is just, okay, what is our brain? Well, our brain is this chunk of matter that sits at the top of our of our bodies, and it's been there for about 120,000 years in its current form. Of course, the brain is much older as an evolving thing. I mean, the mammalian brain is perhaps, say, 100 million years or 200 million years old in a recognizable form. But the cortex of Homo sapiens is not quite that old. There are a number of ways of looking at it, just as a brain, and people have done this for a while. And for example, two very well-known ways of approaching it are to describe it in terms of a functional abstraction. This is where you just basically describe which different regions of the brain seem to be responsible for behavior and function in a specific area, in, say, for example, language or motor activity or visual system. The other is an abstraction in terms of how has the brain evolved, what different parts of the brain are responsible for significantly different of operations. And then, for instance, you'll have the brainstem, which is really the oldest part of the brain and resembles closely the reptilian brain, which is responsible for reflexive behavior. And then the limbic brain, which is a little bit younger, but you can find this also in many other species that are not mammalian, and has an emotive importance. But this is perhaps understating its importance, because even though these older regions of the brain seem to have processes that are not reasoned in the sense that we understand it from what the neocortex is about, how the mammalian neocortex operates and functions, they're closely related to all the functioning in the higher levels of the brain, and absolutely necessary. For instance, one of the components that I've studied a lot in the limbic brain is the hippocampus, and the hippocampus is absolutely essential if you want to be able to store new episodic memories. And episodic memories are completely essential if you want to be able to understand causal relationships between things, which you need to deduce if you want to be able to do reasoning. So all of these older parts of the brain tie into what we consider the neocortex, and that's an important thing to understand when we go forward and look at the types of modeling that are being done and the systems of the brain that are being investigated. We can't just say, oh, let's model neocortical columns, and this is going to tell us everything about the brain. All of these different modular components are important for a reason. Now, there are about 100 billion neurons in the human brain, and somewhere between 150 to a quadrillion synapses, depending exactly how you count it and what you believe. The firing rate is anywhere between 20 and 1,000 hertz, though I have to say that 1,000 hertz is extremely high. That's the absolute maximum rate that you can find among any neurons in the brain, and perhaps only for very short periods of time. The more reasonable interval is between, say, 20 and 100 hertz. Okay, so since we're talking about the scientific origins here that lead towards asim, or Advancing Substrate Independent Minds, a quick look at one of the big projects out there, this has to be mentioned every time, is Markram's synthetic neurophysiological neocortical column. It's the project known as the Blue Brain Project. In this case, what Markram is trying to do is to reverse engineer the human brain, or at least, in this case, the neocortical columns, to a molecular level. It involves very detailed simulations of the structure, the neuroanatomy, and how this affects the function. So these are compartmental neuronal models that he's making. They're really looking at the detail of how the models are affected by their structure, by their neuroanatomy. And it involves short-range connectivity and long-range connectivity that are derived from statistical evaluations of the short-range and long-range connectivity found in mammalian brains, mostly rodent, but now also deriving some information from primate brains. So we'll see where that goes. The model that has actually been built here in this case is a model of, let's see, he's working towards 60,000 neurons, but right now I think the functioning one is a 10,000 neuron model. I'm not sure how close he is to the 60,000 neuron model. What he really does right now is that they're running this model on one of the blue gene systems that IBM has supplied, and they're just keeping it as a running model where postdocs and PhD students can come along and they can do experiments with it. They can design experiments in which perhaps they try a certain stimulus that they'll give to a certain part of this neocortical column, or they'll examine something about the emergent behavior, the emergent activity. But basically they just keep it as a running model in that computer. Just one word about this though. This is a composite of a lot of statistical information. So that's something to keep in mind as we go forward. Another model, another synthetic model, Itzikevich large-scale models with spiking microcircuitry. Eugene Itzikevich, of course, is well known for his neuron model. He's developed a very nice model of a neuron in which you can replicate the response functions of a variety of different naturally occurring neurons without having to care too much about what's going on at the channel level. But he has also made some remarkable studies with large-scale neural models. And one of his well-known models is a model of 100 billion neurons, meaning the scale of the human brain, with a quadrillion synapses, which is sort of the upper limit of the number of synapses. And in that, he's made some interesting observations. He's observed that if he's put this together using a set of microcircuitry that he's derived from knowledge about what the microcircuitry in parts of the brain are, that he can observe emergent rhythms like alpha rhythm and gamma rhythm, moving clusters of patterns of activity throughout the brain, which look a lot like the sort of activity that you can see moving about if you do an fMRI and you watch what's going on in a human brain at a large scale. He's done a simulation which took 50 days to get 1 second of simulated activity out of this model. But of course, he didn't have a blue gene computer to work with, so I guess you have to forgive him for that. There were some shortcuts taken with this model that should be mentioned, which is that, at least in the version that was run there, he didn't actually store any of the synaptic weights. So the synaptic connectivity was recreated on the fly every time that another step was taken in simulation. There we go. Now, just as a summary, kind of what all of this leads to, these types of simulation models, is that you can build something like a virtual laboratory, virtual brain laboratory. That's very interesting, not just for neuroscientists, it's interesting for neurosurgeons, for example. It's interesting for pharmaceutical companies, engineers, AGI researchers, because you can do things with a virtual brain laboratory like that. You can test things before you go and take them to any kind of clinical trials or take them to animal trials. And okay, maybe it seems like you have to do that anyway. You need to go to the animal trials and the clinical trials because otherwise you'll never be allowed to do anything or make any statements about any new discoveries you've made unless you've really tested them on something that's validatably a real brain. But it does mean that you can test a lot of things before you make that step. It's sort of like the advance that came along when the aerospace engineers no longer had to rely on just building prototypes and testing their prototype in a wind tunnel, but they could do a lot of their tests on computers first, which means they were able to go through 50 different possible wing shapes before they tested one in a wind tunnel. And this is the sort of advantage that you get when you do things virtually, when you do things inside a computer. So models are really interesting to science, to engineering, to pharmacology, etc. Now, there are other origins as well that overlap with what asim is about. For example, there's an integration coming from AI and AGI, AGI being artificial general intelligence. There's a growing interest in looking at the brain as a system that could tell us how you would make an intelligent system. And I don't mean that just in the general form where you say, oh, look, it learns, but how does it learn? How does it do something? There are some good reasons why you would want to emulate such a biological brain rather than just coming up with a de novo architecture. The main reason, of course, is that this is a system that we know works. There's evidence all around us. That means that everything that we try with a system like that is properly grounded. We can ground our assumptions, our hypotheses about how do you do something intelligent? How do you really recognize an object and make this association with what it means and what you can do with it and what it has meant for you in the past? An example of an attempt to use emulation of what's going on, emulation, perhaps simulation is still a good word for this, in the brain is the project that Dharmendra Modra is carrying out with IBM. Again, IBM is involved. And in this case, it's in the context of the DARPA Synapse grant, which was for systems of neuromorphic adaptive plastic scalable electronics. In fact, that's really the target that Modra is going after himself. He sees this as an engineering challenge more than a scientific challenge. He's interested in what can we do by simulating to great detail what is going on inside natural biological cortices, and how can we use that to make interesting new circuitry that could do something that we haven't previously been able to do by eventually putting it into hardware, neuromorphic hardware. So his approach is more uniform in the sense because he's interested in it as a template for the building of new types of circuits. It's not so much the approach that Marker is taking. And this led him into a bit of a controversy because he was very happy to declare that they had done a really large simulation of something that is, it's based on this more engineering approach, but it was large and it had the processing power, let's just say, of what is assumed to be the power necessary to run something at the scale of a cat brain. And this got him into some trouble, of course, because it's not quite the same approach that Henry Markram is taking. But now it's pretty clear where those two are headed, and they're different directions. Yeah, and then just finally, I should mention that what you, of course, can aim for if you're doing whole brain emulation, or what we want to do for substrate independent minds, is that you can aim at human-derived AGI. So human-derived AGI really comes in two forms. It's either basically emulate how a human brain works, and that is your AGI, or learn as much as you can from human brains and use this as your inspiration for how to make AGI, rather than just try and come up with a completely new algorithm for what is intelligence. Not that those aren't interesting, but this is where it overlaps with what we're doing. Okay, and then of course, another origin which can't leave out, which is neuroprostheses and augmentation. Mostly we go back here to the work that's been done in BCI, brain-computer interfaces, also known as brain-machine interfaces, BMI. There are a number of different kinds of BCI. I'll just go over them very quickly, but there are non-invasive kinds, EEG, MEG, fMRI, NIR, and semi-invasive BCI, like electrocorticography, which means that you don't put the electrodes on the surface of the head, but you put them on the surface of the cortex. That's, I think I should make this distinction slightly more clear, which is that in non-invasive BCI, because you're sitting on the outside, you have to kind of cope with the fact that you're either going to get low spatial resolution, or you're going to get low temporal resolution, or even both. Both problems may occur, and you can sort of improve your reception of the signals in the brain, obviously, if you take off the cap and you get closer to the brain. So if you put the electrodes right on the surface of the cortex, and this is done, for instance, when trying to treat epilepsy, they like to put these grids on the cortex in order to discover where exactly are these seizures coming from before surgery is carried out to take care of serious epilepsy. Now, when that's happening, then you can also go in there and you can use this as an opportunity to do some scientific tests. And some people have in fact done that, like Mike Kahana and Mark Howard. They've published very interesting papers showing that you can detect very specific increases in rhythmic activity at gamma and theta frequencies. The power, you can see how it correlates directly with work done on remembering lists, for instance, lists with different lengths. And this ties right into developing validatable models of how those systems work. Now, and then finally, there's invasive BCI, which is where you really want to get at the high resolution information in terms of at least specifying that you're reading from very specific neurons. You're going for 1, 2, 3, maybe a few more neurons, sometimes maybe a few hundred if you have a very large set of invasive wires, and you can get high temporal resolution from this. So invasive BCI, this is the typical approach when you're either studying from animal studies, when you're doing that, or in vivo, or if you're going for some kind of interface that you're trying to provide in a prosthetic way for people who need to get out of a locked-in state and control the cursor on a screen or something like that. But you can also try non-invasive approaches for the same reasons, at least in humans. Okay. There were more, yes, of course. Sensory prostheses like cochlear implants and retinal implants, deep brain stimulation, vagus nerve stimulation. It's a bit of a hot topic right now. Transcranial magnetic stimulation. This isn't really as neuroprosthetic, perhaps, but it has some of the same uses. Okay, let's carry on because I don't want to spend too much time on that. Just one that I'd like to point out because it's very interesting to us, which is Ted Berger's cognitive prosthesis. It's interesting for two reasons. One is that it's daring. It's the thought, okay, let's just not stick with only sensory prosthetics, but why not go for a cognitive neuroprosthetic, something that actually replaces a piece of the brain, of the internals of the brain. But it's also interesting to us because Ted Berger, in his approach, quite clearly is aiming for something very similar to what we're talking about with substrate independent minds. He's saying what we're doing in there, what's happening in this piece of the brain that he's looking at, does not have to be run on biological hardware. We could run it on something else and it would work just as well, or it would work well enough to replace the lost function. And in this case, the function we're talking about that he was looking at first was the function of a small part of the medial temporal lobes. He was looking at the area CA3 in the hippocampus. That was his first target. He wants to move it to other targets now, like the neocortex. That could be difficult. And there's a reason why I think that could be difficult. The hippocampus carries out a very specific function where perhaps it's not so important what the individual connectivity is for one specific subject in one specific subject's brain, just because it has to line up new information that's coming in and prepare it for eventual storage somewhere else. So this is a temporary storage area. It has sort of a buffer function. And that I can tell you can do with an approach such as Ted Berger is doing, which is to create hardware based on data collected from numerous different subjects. In this case, they were most rodents, now moving into primates, instead of from a specific individual, because you don't need the individual structure, you don't need the individual function there. You need what does this thing do in general. But if you're going to move to the neocortex, well, then you're going to run into some problems, because the neocortex is a long-term storage area. It's a place where perhaps you're storing the memories of, I don't know, the people you know. And if you lose that information because something's damaged, because there was a tumor and it destroyed some of that area, then putting in a general circuit, it may replace what that area is supposed to do, but it's not going to give you those memories back. We have to remember that the brain, though it's a distributed system, and it distributes its information over many different neurons, and a lot of the neurons can disappear and most of the information will still be there. The distribution is not global, it's still a modular system. So it distributes its function among many neurons within a certain area that carry out a certain function. Okay, so now a little bit about need. Why would we want to? What is the need? What kind of external causes are there and trends that would underscore a need for advancing substrate independent minds? Well, okay, the brain is a wonderful thing, and let's not forget that. But there are also some computational limits to what human brains can do. Short-term memory, for example, is quite limited. Whether you believe in the 4 plus or minus 1 or 7 plus or minus 1, the exact constraints depend on the modality, they depend on the region and on the task. But it certainly is constrained. Another example is long-term memory, which is clearly finite and fallible. I mean, I can hardly remember what happened yesterday. Honestly, if you ask me between 10 and 11, list what I was doing, impossible. So clearly long-term memory is, it's not great at uptake of information, and it's also not so great at storing information. Processing rate and multitasking limitations. Well, for instance, I can't really do 2 conversations at the same time. This is very difficult for me, so I'm limited by modality. Within one modality, I can really only do one thing at a time. There's no multitasking there. And my processing rate is also quite slow for many types of tasks. There's bounded rationality. Take, for instance, our tendency not to estimate objectively, but instead to either completely optimistically overestimate how well something's going to go, or to be way too pessimistic. And if you want to see examples of this, just look at what goes on in the stock market as everyone rallies or everyone pulls back, and it's usually a complete overreaction to what's going on, but it's very typical of human behavior. Sparse creativity, which is perhaps a bit controversial, but it could be said that while we are creative species, we're the most creative species there is on this planet at least, but but how often do we really apply creativity to what we do? How often do we simply do what we know, or just do what someone else tells us? How often do we actually sit down and come up with something new in a given day, in a week, in a month? You know, creativity perhaps could be enhanced further. Then there are limits to our ability to pay attention to things. We really can say that while short-term memory has a limit of, say, 4 plus or minus 1, attention and consciousness, this is really limited to 1. That's the span, attention to one thing. Maybe there are some weird occasions where you can pay attention to 2 things at the same time, but it's more like taking turns between them. Limits of comprehension and sensory experience. This, of course, well, I mean, you can see that all around you. There are animals that can sense infrared. There are animals that can sense magnetic fields. There are animals that can sense various things that we can't do. And then, of course, there are machines that can detect things that we cannot even see, cannot measure, cannot really experience. And it's not just a matter of sensory experience, which is limited, but it's also what we can comprehend. How can our brains process things? the fact that our minds are Turing machines, or that I think that they're equivalent to a kind of Turing machine, means that they are also limited to what Turing machines can carry out. So just to summarize that bit about limits, we really only have 100,000 years of human brain development and experience. And this is out of 13 billion years in which the universe has existed. So not only does this mean that we have not really gone as far as we possibly can, but it also means that, say, for instance, if we ever were to meet another intelligent species, the chances would be that they are far more advanced than we are, simply because, if you look at the curve from 0 to 13 billion years, the chance that they've been developed, that they've been going through much more development, much longer time, much greater. So that's just, this is something to say. Okay. Okay, more about needs. Competition and adaptability. I think that competition is an inescapable occurrence in the animate and in the inanimate universe. And I say this over and over again because I think it's important to understand. To give our minds the flexibility to transfer and to operate in different substrates bestows upon our species the most important competitive advantage. This is something to think about for a moment. I mean, the environments that we operate in right now are very severely limited. We can only live basically on the surface of the Earth in this biosphere. And that means that as a species, we're subject to various risks based on that. And we're not really easily able to go out and live anywhere else. And we're not easily able to compete with other types of thinking, other types of behavior that may come along. So in the long term, natural selection favors the most rapidly adapting and improvable. Okay, you can contest this, you can contest this if you like, but since obviously there are also species that still exist which are very primitive, but in our case of fairly large, complex organisms, it seems that being able to adapt quickly is one of the important factors in survival. So I think that this is something to consider both before and after a potential singularity, if this is what you think we're headed for, that competition will still be an issue. Competition and adaptability are always issues. The question is, if we ever develop artificial general intelligence, should we just, why should we just let them benefit from all the possible upgrades and rapid development that they can go through? Should we just sit back and let them take off and start to discuss things that we can't even understand? Or would we want to be able to participate in that and understand it as well? Do we want to be able to adapt to the levels that AGI could adapt to? Do we want to be able to compete with them? Do we want to share information with them? These are all things that come into the needs category. And then there's this matter of purpose. Well, I don't know, maybe I shouldn't even go into that, but I think that there can be no universal purpose. So there isn't any global reward that we can assign that we need to optimize towards. But we can define local purposes. So when you decide to go for something like substrate independent minds, it means that you're choosing to take part in a future where you can still compete, where you can still adapt. It also means that you're seeking out uniquely novel experiences because, for instance, bringing in new senses, bringing in new comprehension, these are ways of expanding our experience as we have it now. But it's really a choice. It's a choice that you want to continue to acquire new knowledge, that you want to continue to do something with that knowledge in a time when maybe technology has moved on past a point where the human brain as it is right now can understand what's going on in there. Then there's the issue of life extension. This is also another area that overlaps with what substrate independent minds is about. Because, of course, if you can move to another substrate, then it may be that you can continue moving to substrates and dispersing the risk that you somehow end your existence. This is different from in-place biological life extension ideas, which are abound, and of course are quite naturally the first thing people think about. But any one of those can only get you so far. At some point something happens, and you can no longer rely on, let's say, a biological defense mechanism or a biological repair mechanism to keep you going. Now, what sort of things does substrate independence give you? What could you look forward to if we can achieve this? Well, one thing is that you can have both a physical and a virtual existence. You can exist in different types of realms. You could travel by transferring around or existing in a cloud of computation, let's say. You could augment your senses and your cognition. This is something we just talked about before. Understand physics in a way that we never have so far. And continue to be able to converse with and share in the development of intelligences out there. Okay, so now I've talked long enough about the why and what is it, and now let's get into the real nitty-gritty about how would we do this. And that's important because this group and also the new organization carboncopies.org is really about how do you get something done. Practical approach, active approach. So first of all, why consider the whole brain? Well, a really important reason is validation. The whole brain is the one implementation of mind that is known to work. It's the same argument as for AGI, which is that, okay, here we have something, and we know it works. So let's look at that first. And in this case, it's the specific architecture, the specific functionality of what makes the brain that we know works. It can cause an emergent brain, cause an emergent mind, cause all of the functions of mind to emerge. It's a modular architecture, so there are dependencies between all these different modules, and that's why whole brain may be absolutely necessary. In fact, there may be a dependency on things that go beyond our brain. There could be a dependency on our peripheral nervous system, a dependency on the sensory input from our environment, the way we're embodied, the way we exist within our environment. So actually, even just looking at the whole brain is already putting some constraints on it, and we may need to look further. So what are the significant parts of the mind? Okay, I'm listing a few things here, but say, for instance, new episodic memory, you want to be able to continue to generate new episodic memory. These are functions you want to be able to take on, and this is a part of the modular brain. It's part of the limbic system, for instance. It's part of those different shells, let's say, those different parts of this onion that really makes up the brain. There are inside the functionality, inside the structure and the function of the brain, there can be hidden characteristic functions. This is an issue if you're only looking at the behavior of the brain, as many would like to, because it's the easier data to access. If you only look at what's coming out, then you could be missing potential very important characteristics of how that mind functions, because you don't know the kind of activity, the kind of responses that it could create given some other input, given something that you didn't happen to observe during the experiment, or didn't happen to observe during the period where you were life logging, let's just say. So these are important things to consider about what's going on inside that brain. And another reason why we would consider the whole brain is it's also purely practically that a lot of the methods that we're actually investigating, the methods that we would like to be able to investigate, are methods that you can apply equally well to some small part of the brain, to the brains of very small animals, or that you could apply to a whole human brain. It's just a matter of scaling up. It's not really a fundamentally different technology or a fundamentally different approach. So in that case, why not consider the whole brain? Right, and just a few quick words about some fundamental concepts here to try to explain some of the things that we need to think about in order to understand what it takes to emulate brain function in another substrate. We need to somehow define what it is that a mind actually does, or how do we look at a mind and say what is going on in this? How do we describe the function? So I'd like to first of all give it what I call the zeroth order quantification, the mind state, which is just, okay, we want to be able to say something about what's happening at time t. How do we define what the mind is? We could take a snapshot of it. We could say, these are all the connections that are there, and this is, I don't know, this is how it's, going to behave in the future because of that. It's kind of going on to the next step, but really a snapshot means what is it that has elicited whatever awareness or behavior you have right now? Can you take some kind of representation of that so that you can use it to develop representations of what is in the mind at future times. So that brings us to the first order quantification, which is what are the transfer functions? How can we say at time t plus delta t, what is the state of the mind going to be given a state at time t? We need to be able to predict that if we want to do an emulation. We need to be able to predict it well enough with an acceptable variation from what it would have done, what would have been the state at time t plus delta t if we were using a biological brain. This means we want to talk about small dt's, small span of time, and we want to be able to talk about enough detail to make those predictions. We can express all of this in control theory terminology, and this is in fact something that Elias Smith has already done for dynamic neural networks. And here we can try to apply this at a number of different levels of description. I'll get into the levels of description in a moment. Just one more thing about quantification, though, because transfer or transition functions, they were not the only thing you have to look at. I mean, remember, we're talking about a brain that develops, a brain that learns. So the transition functions don't stay the same. If we're talking about how did our mind function when we were 5 years old versus how does it function now, clearly a lot has happened between then and now. And there's a reason why a lot has happened, and that's because there is this change of function. We need to describe that as well. That's the second order quantification. All of this together gives us a bunch of parameters, a bunch of structures, and how they modify over time. So what is the minimum data that retains information, or the information theoretic mind? What is it that we would need to capture? We need to look at the brain at a sufficient spatiotemporal resolution, and we can think, for example, these are just examples of levels like localized brain activity. What is it that occurs in certain voxels of the brain? You can think of, for instance, what fMRI gives us when we look at the brain. The activity of groups of neurons. Groups of neurons are responsible for certain subfunctions. They work together. No neuron does things all by itself, or hardly any neuron does things all by itself. It's always groups of neurons. So perhaps groups of neurons are the right place to look. Time averaged, or rate activity of individual neurons. Well, this has been thought to be the relevant issue for a long time. I mean, we know now that there are things that go beyond rate, but rate does seem to be very important if you just look at the way that the brain works, and everything is rhythmic. Everything has different rates of activity. Or perhaps we should be looking at the spiking activity of individual neurons. It's now known that there are functions carried out in different species that can only happen at the rate, at the speed that they're occurring, if the brain is able to react to a single spike of a neuron, rather than, or a single spike of a set of neurons that happen to be fairly well synchronized, not a rate that needs to be measured over a period of time. So in this case, it's the phase at which activity occurs that's important. So maybe this is the level where we need to look. Maybe that's what we need to describe in our control theory view of how the brain works. Or maybe we need to look at the analog membrane responses. Perhaps it's important to understand how all of these changes in the voltage, let's say, in the potential inside the cells, how that affects how the neuron is going to react, how it's going to act in the future. Or maybe we need to understand all of this, not just in terms of specific cell locations or areas or regions of groups of neurons, but we need to understand this throughout the architecture, the neuroarchitecture, the neuroanatomy of individual neurons. So look at what's going on at the ends of dendrites somewhere. There are specific spine locations and terminal locations. If we need to understand how the activity is propagating through those branches and being interfered with by other activity that's occurring at another point on that dendritic or axonal branch. Maybe we need to know all of those details as well. Maybe we need to capture all of those. Or perhaps you need to go even lower and look at responses that are happening at the molecular level. So we can consider all these different resolutions and wonder which one do you need to look at if you want to capture the essence of information needed if we were to transfer function of the mind to another substrate. And of course, these resolution requirements can differ depending on whether you're looking at state, transition, or update. Then there's the matter of function and structure. At each one of these resolutions, information is captured in the structure. How do different components interact with one another? Who do they communicate with? It's also captured in the function, of course. So what is the response function of the individual components? Both of those are significant to some degree, and we know that we can infer things by looking at one or the other. We can infer something about function by looking at structure. Structure can help us discover hidden function, for instance. I was talking about the hidden functions in the brain before. Well, if you're observing the function of a part of the brain over a period of time, there are a number of things you can learn from it and you can infer. For instance, you can use function to infer structure. You can infer a lot of the functional connectivity that way. But there are some things you may be missing because there was no activity to give you evidence that this could go on in that piece of the brain. Whereas if you have the structure, if you get the structural information, then you could infer, oh yes, these connections exist, so that kind of response would occur given such stimulus. There are things that you can infer about the function from the structure and vice versa. Good. So, what are the kinds of targets that we could go for? What are the possible target substrates if we want to do enough implementation of mind? And by targets, I mean a number of different things. One, of course, is the computational hardware that we're thinking about. What is carrying out these brain functions? And the other is embodiment. So how does it interact with the world around it, or where does information come in and go out? The computational hardware, well, let's just go from really re-implementation in biological form, first of all. Talk about that because that would be the immediate thought, of course, of someone who's saying, okay, we want to re-implement what's going on in the brain somewhere else. We want to transfer it. Well, why don't we just use also the same components to make this possible? If we're already looking at the biological components to try to understand what's going on in there, why don't we just re-implement again in those same types of biological components? Sure, we have to consider that. That's a possibility. Nevertheless, probably the most common way to think about re-implementation is in terms of software on general purpose computing hardware. This is because we're used to modeling things that way. We're used to doing simulations and emulations in software. It's easy to handle. You can quickly modify it, and we have all the general purpose hardware available. Another possible target would be neuromorphic hardware. The thought being that why not create something that operates in a similar manner as the biological components so that it is well matched to the necessary functions and thereby makes it possible to more easily transfer from the one to the other. And then there are various exotica that you can think of that might help us both to be able to run these emulations and to expand ourselves and go towards new possibilities. Then in the case of embodiment, I think that, oh, I see, just someone is typing about possible target substrates. Very interesting. Okay, I think that because I'm not able to pay a lot of attention to the text chat that's going on, if people really have questions or want to discuss something specific about a slide, then maybe they'll have to open up the mic for a second and say something. I just want to throw that out there. I'm not really paying attention to the text chat. Okay, in terms of embodiment, well, the question is what happens when you actually emulate a mind, when you put a mind out there into, let's say it's in a computer. The first thing you could do is simply sensory deprivation. Just have an emulation and don't bother with what kind of embodiment you give it. I know that sounds wrong right off the bat because we've already been thinking through what it might cause, but that is, of course, the first thing you can do. You just have the emulation and it's there, and it's sensory deprived. And what happens? Does it start dreaming? Does it go crazy? I don't know. That's the first thing you can do about embodiment. Another thing you can do is try to give it virtual sensory motor environments. You could basically put it into Second Life or something like that and hope that this is a good place for an emulated mind to exist. You could try biological re-embodiment. This is kind of on the same train of thought as the biological reimplementation of the computing hardware. A lot of people would probably like to see this as the way to go with substrate independent minds, that yes, you can move it out of this brain when there's something wrong with it. Let's say you're getting Alzheimer's disease or something like that, but then you can move into a new brain and a new body, and you go on living as you were used to. Or you could physically embody in other substrates. You can have, let's say, some part of yourself is existing within a virtual environment in a cloud of computation, and another part of yourself is going out there with some kind of robotic body flying off to Mars to explore the surface or something like that. These are all possibilities to think about. And that kind of leads me straight into a few other interesting possibilities, and also into this discussion of how do you get there? How do you get to these different substrates? Well, one way to do the transition is to look at what kind of change is already occurring around us, the context in which all of this is taking place. There is augmented reality that's coming along, so we're becoming more and more dependent on technology just to understand the environment around us. For instance, I'm getting so used to the GPS device in the car that, this is just part of how I drive. It's part of my driving capability. It's a bit of my brain as well. Then there's augmented cognition, such as what would it be like to have a quantum computing brain and novel senses, and what can you do with that? Or you have here a picture of a life logger. This is someone who doesn't just want to depend on his own memory to store what's going on around him, but instead is relying on other technology that makes real, verifiable recordings of what's going on around him. So these gradual transitions toward, I guess, what you would call cyborgism, slowly changing our dependence on machines, our dependence on living very intimately with computing hardware, this is going on. This is something that already slowly brings us to a kind of different being, a way of having parts of our minds already outside of what we consider the biological brain. The lifelog issue brings us to another question about transition methods. How can you get that data extracted out of the mind, out of the brain, and bring it to another substrate? Would it be sufficient, for example, to do a lifelog? Could you use that to capture all the data? This is a topic that comes up, for instance, in connection with projects such as CyberRev, which is where the the Bainbridge-Rothblatt method is what we call this. You can see as, okay, let's just collect as much data as we can about a person in all sorts of different formats and use it to try to represent what this person is or was in another substrate. Whether that technique is developed enough sufficiently to do what we would like to do with it, if you really want substrate independent minds, if you want to be able to transition a mind out of our current biological brain, that's really debatable. But the thought that it would be possible to at least describe what is a human mind in general terms and then in specific terms for an individual, that you can separate those somehow. There's nothing really controversial about that. After all, we're talking about data. We're talking about a machine that processes data. And we're talking about an instantiation of a certain amount of information. There's nothing unique about this. This is something that you can do with any kind of data. So really, looking at means, for example, this is just a statistical technique. So there's nothing really controversial about the idea that we could at least describe what are people in general. We've been recently calling this a me program, this idea of, okay, that's a person. Take that data. It's sort of the mean of what everybody has in their brains, in their minds. And then what is the difference between those? Where's the standard deviation? How much is that standard deviation, by the way? Would it even make sense to try to make representations of the mean if it turns out that the standard deviation is so large that you don't really gain anything by having those two separate? In fact, if you're trying to describe what is the mean and you're trying to describe what is the difference between individual persons, do those descriptions are they properly done using the same bases? Can you describe them in the same system so that you can easily combine the two? Those are all still things that are really not so clear. And in fact, it's quite possible that to get an understanding of this and to be able to use this approach, you would need to do the detailed emulation of a brain first. You would need to go through all the steps that we're talking about in Advancing Substrate Independent Minds first, before you can do this as kind of an optimization procedure, where you say, let's abstract out the one, abstract out the other, and then we can have a simpler representation of what individual persons are and how you can put them together and stuff like that. So it may be that while this looks easy to begin with, it's actually something that depends on all of the other developments that we're talking about. Okay, so one of the techniques for transition that has been explored by several, and here I mentioned Denk, Lichtman, Hayworth, and Yoon-Suk Cho, is the extraction of post-mortem data, structural data, from getting large-scale, high-resolution neuron anatomical structure. There are different methods here, and there are devices that have been built for it. Just to mention two, the automatic tape collecting, the automatic tape collecting lathe ultramicrotome. Am I saying that right? Anyway, and the knife edge scanning microscope. So these methods rely on the fact that we can at least store our structural information by plasticizing brains, or by in some other way creating a static a brain bank, a place where you would put a brain, and then you can just use it as the original data. You can use it by slicing it up, by storing those slices perhaps, at least this is what the Lichtman method does, and then analyzing those with a microscope and reading off what is the structural information, reconstructing from the images into a representation in 3 dimensions, and from that inferring the function by saying, in these 3 dimensions we recognize components A to Z, and they operate in such a manner, so we can recreate a functional version of this brain based on the correlation between these known component functions and the structure that's been identified in these portions of someone's brain. That's fine, and the good thing is that this is a technology that can be pursued immediately. There is already technology out there that can help us to achieve this. There is the issue to consider, though, that it's inference from structure about function that we don't really know enough about yet. We don't know about the function of all these different components, and we don't in fact know how all of them together would operate, and how this may differ from what we think that they operate together if we just put all the components together in the manner that we understand them right now. So there's a matter of testability. If you have a brain that has been plasticized, and then you go and infer its function from the structure, you can't go back. You can't go back and test, how did this brain operate? How did these neurons really work together when it was alive? So there's this issue of testability and validation, and of course the quality of such an emulation depends on our real understanding of what the components do and how we infer component function from the structure. Okay, then there are in vivo recording methods. The most common method to get in vivo recordings from multiple neurons, from networks of neurons, is microelectrode arrays. Peter Passaro was kind enough to talk to us recently about how to do this in Limnia. And it's very promising work in that there's the ability to extract functional connectivity, as you see it from the activity going on in the network, to understand how each neuron is working together with the other neurons around it. So we're not just getting single cell recordings, we're not just getting a very artificial view of how the neuron operates by itself, but we're getting a view of how it interacts with its neighbors and how functions are getting done. And in this case, functions of specific parts of the brain of Limnia and what it carries out. This is promising, and I think that the most promising route is in fact to integrate what we're learning from this in vivo research and what we're learning from structural research. So how can you take this functional information and use it to help put together a correct emulation of what's going on by using the structural information as your basis to infer the hidden functionality that you did not observe, those functional connectivities, those actions and responses that you couldn't observe during your experiment. Okay, these are the technologies that we have available right now for the kind of study that I think will be necessary to properly understand how to transfer mind to another substrate. But what we really want is we want to go even beyond this. We want to be able to use novel technology to really access information at large scale and high resolution. I'm going to talk about that some more towards the end of the talk, but just to mention a few of these technologies coming up, synthetic biology is going to be emerging in the next few years as a very viable technique to investigate what is going on inside our bodies and inside our brains. We can already see the beginnings of that through optogenetics, in which viruses are used to deliver specific modifications, let's just say, to particular cells that are targeted to now react to light in different manners, sometimes to inhibit or to excite activity in those neurons. So you can selectively target areas in the brain, you can selectively target specific types of neurons to test what happens if you activate or deactivate them. This is just one example of where we're going with what I would call the whole area of synthetic biology and how it can affect research as well as development in these technologies. Then there's the consideration that the field of AGI, which already has a good reason to be interested in what we're talking about, can itself help us achieve the technology that we need to move minds to substrate independence. Because there is a lot to explore. This is a huge field with a lot of different possibilities. In fact, it's kind of like a combinatorial explosion of things to research. But AGI can help us to do a lot of the grunt work there. I think it can explore many possibilities more quickly than we could, or better than we could, if properly guided, if properly developed. So I think this is an area where there has to be a dialogue in both directions. AGI is interested in how what we learn about the brain can be useful to make AGI, and we can learn how to use AGI to get us to our goals. So that's sort of the cutting edge stuff. And I just want to, as a final point on there, that I think that though the structural and the functional, they both have to exist in one form or another. We shouldn't forget that if you have a procedure where we can gradually investigate the functionality of what's going on inside particular portions of the brain and transfer them to another substrate as we're doing this investigation, as we study what's going on, that's really the most cautious approach for how to transfer a mind to another substrate, is that you observe which changes you're carrying out as you're doing them and whether there is any fallout from that. Okay, I might have to start going a little bit faster because I feel like I've been talking for a long time. So I'm going to go through this a bit quickly. So discovering the resolution, what are the using regional significance limits. This is the idea that the brain, because it is modular, has different areas that are doing different things, and they process different kinds of information. So for instance, you can use knowledge about the type of information that's coming in and its limitations, say its bit rate, its quality, its resolution, to infer what kind of resolution may be necessary in that portion of the brain if you want to represent it in another substrate. That's something that can help you with the resolution problem. And I think that's kind of what I just said here, so we'll just move right on. Yeah, one little word about why consider in vivo. Okay, well, I've already done that, so I'll go past that as well. Okay, why combine large scale and high resolution? Just one more slide about that, even though I think I've said it before. If you're just looking at low resolution, large scale data, like fMRI, for example, then during that experiment, you're not really exposing yourself to many of the existing functions that are being carried out by the brain. Again, we've got this limited inference that we can make. It's kind of the same problem that you have in many of those techniques. On the other hand, if you're only looking at high resolution but small scale, say the multi-electrode approach, but in some parts of a human brain, take that scale and just put in a few multi-electrodes in various places, then you have a very limited understanding of what's really going on in the entire brain because you're only sampling. So we want to combine both. But in vivo is still lagging behind a bit, and really what we would want to be able to do is to, let's see, go next list here. We'd want to be able to make measurements that record episodes of what's going on. We want to be able to do measurements that are very physiological and morphological, that give us information about both of those. We want to be able to do it over a large area of the brain, preferably the entire brain, with many recording sites, to observe how the different parts of the brain, as well as local groups, collaborate, and how the neuronal clusters really are responsible for different patterns of activity in the brain. We want to be able to look at this at high resolution and high rate. Some operations are sensitive to the activity of individual neurons at specific times. Now, the existing in vivo interfaces, they lack spatial and temporal resolution, and it's often not possible to even identify the kind of information we're looking for. Let's say in imaging, for example, let's say fMRI, you can only see a certain type of information. You can see the uptake of oxygen, for example, and that's not necessarily what you need to know, because there are other kinds of activity going on there. So really, we are kind of at a point where we need new tools. Just a quick list. I don't even know if I need to go through it all, but you can read it off of the slide. Some of the aims, if you want to do good in vivo recording, the next generation of in vivo recording, of course, safety, like it has to be harmless, low impact preferably. This is not so important when you're just doing studies, but if you want to do this with human subjects, at some point it becomes absolutely essential. The performance requirements, well, we need it to be reliable. We need to be able to do large scale. We need to be able to go and do high rate data acquisition. This is all fairly straightforward. But these are very important developments that current technology just can't handle yet. Okay, I won't go into that right now. I'm trying to skip through a little bit. Okay, this is just an idea for a future large-scale, high-resolution method of detecting information. Yes, is there someone on the microphone? Okay, that's not a question coming up. So I'll just move on past this. I don't really want to talk about nanoscale agents at this moment. That's really talk. Engineer biology. Okay, we're reaching the summary of the talk. What I really wanted to focus on right now is just to say a few important things. Okay, we need to do more experiments. We need to demonstrate and analyze. Basically, what we need is to discover the resolution requirements. What are going? This is going back to the fundamentals of what do we need to know about the brain if we want to move the mind from one substrate to another. What we also need is we need good proofs of concept. Basically, we need some experiments and technologies that demonstrate that this is possible, so that there's an understanding of, oh, okay, yes, we can regard what's going on in the mind as processes that are carried out in a machine, whether that machine is biological or not, and we can move this to another machine. So this could be a matter of showing that you can do it with a very small brain, that you could do it with a part of a brain, that you can do it somehow. There has to be a proof of concept, and I think that many technological paths will contribute to this. This is a convergence of many things. A lot of projects can focus on what they're doing, of course, on what their specific target is, but I think overall, to demonstrate that we can achieve substrate independent minds, we'll need to converge. We'll need to bring a lot of things together. Then finally, with all of this activity going on in the context of developments that are happening everywhere, such as augmentation, such as the structural analysis of the brain, such as new computing hardware being developed, and everything that's happening around us that is related to the objectives that we have, it's important to continue to see the forest through the trees. So we always need to look at the results coming out of these projects and the results coming out of what's going on around us to understand how well does it satisfy the objectives of advancing substrate independent minds, which I think are something that we're trying to describe in more clarity right now, and then we can keep track of what's going on. So this is really the summary of what I think we need to be doing in the next while. And then finally, I just wanted to tell those of you who don't already know that we have a conference coming up August 16th and 17th at the Hyatt Regency in San Francisco. This is a satellite to the Singularity Summit, and you can see all about it on our webpage, carboncopies.org. And I'd like to thank you all for listening to me. And please, I'd like to open up the floor to discussion at this moment.
I just have a question about the Carbon Copies conference. Do you know if that will be streamed online?
This is something that I've actually been thinking about, and we've been discussing a little bit. We certainly do have some speakers who are appearing online, so we already have a virtual component there. I'm not sure how much more it would take for us to stream all of this. It's very certain, in fact, I think we're going to record everything and make it all available, but whether we can stream live, that's still a question. I'm not quite sure how we would go about that, given our, I guess, our finances and our, what we have available. I'm not sure, Giulio, do you know anything about that?
Yes, this is actually the plan that we're going to stream the conference in San Francisco via Teleplace. The only requirement, we haven't really discussed it so far, so we discuss it in real time. The only requirement is to have a good internet connection in the San Francisco meeting hall. And we have been discussing this on the mailing list, and I understand that there is going to be an internet connection. Now, in case there is no internet connection in the meeting hall, we can do it with a USB key for high-speed 4G internet access, and that's going to work fine. But of course,
okay, so it sounds like you have this covered.
Yeah, but of course, it's much better if we can manage to have a standard high bandwidth internet connection, either on cable, wireless connection. I'm quite sure that the hotel is going to make that available. Now, something that happened to me once when I have organized a similar thing in a hotel is that they did have good internet connection available, but it was outrageously expensive. And what I did, I just used a USB key for internet access for perhaps 5% of the price they were asking. Since I guess in California we can get a good 4th generation USB connection, then it is fine.
Okay, I think we'll manage one way or another, yeah. We'll try to prepare everything in advance properly, of course. We still have about 4 weeks to go on this, and luckily the location is very close to where I am right now, so it's very easy to go down there and test things and try stuff. Oh, I'm seeing there's a question coming up. Could you comment on your estimate of the computations per second to emulate the brain and how it compares to Kurzweil's 10 to the 16? Okay. Well, I can comment on it, sure. I don't know, because as I was trying to point out during the talk, we don't really know the resolution at which we have to work to do a proper emulation. And there's a difference also between research resolution and performance resolution. By that I mean, what are we going to have to look at? How many calculations do we have to do while we're still trying to investigate how this whole thing works? How does a mind work inside a brain, and what does it take to emulate it? Step one, how deep do you have to go then? Once you understand that and you start optimizing what's going on in this emulation, then it's quite possible that you could come up with implementations of the same mind that require far fewer processes, far fewer computations per second to do. So it's like, you've got the proof of concept and you've got the final industrial size operation of how to get minds to work on other substrates. Probably have different requirements. Don't know what those requirements are right now. Don't know if we need to go down to the molecular level, if it's okay to do spiking neurons, or if we go to transform this to a different representation where we're not even talking about neurons. So it's very hard to say. I think that Kurzweil's estimate is, yeah, sure, we can take that, but I don't know what it says really, besides picking one specific favorite target and then saying something about that. But there are a number of these numbers as well, a number of these values in the first whole brain emulation roadmap that was done, and this was based entirely on approach of how do we structurally emulate what's there, structurally copy what's there and then emulate all the function down to certain levels, let's say spiking, or let's say compartmental models, etc. And then it lists the different numbers next to it, so you can also look at that as a reference. I'm sorry, okay, there are other questions going on here? Who asked?
I think,
Randall? This is Tudor.
well, there is someone. Yeah, Tudor, go first.
Julio.
Well, okay, we have been discussing this before, of course, but it's a good. I mean, it's good to have the opportunity to discuss this again. I mean, these are all very exciting and interesting theoretical developments, and technology is a very sexy future technology, interesting for its own sake. But I guess what most people would really like to hear is some kind of a realistic timeline for this development. I do place operational deployment of this technology close to the end of the century myself, but I know that other people are much more optimistic than that, and I would like very much to be wrong. This is my first general question. My second specific question is, regardless of how fast development will actually happen, I think for most people of our generation is probably too late anyway. So do you think, is there anything that we ourselves, the people in this room and other people our age, can actually do to benefit ourselves from this?
Okay, I'll answer both of those questions in turn. So the first question you had is really, what is the time frame that we would talk about to get to transfer to other substrates? And I really have a standard answer for this because I'm asked this all the time. And the standard answer is that the time it takes to get anything done depends on the resources that you put into it. So really it depends on how many people become interested in this. Let's say, for example, we do a good proof of concept and it becomes an immensely interesting area of research, an immensely interesting area of development. Many resources are poured into it. Then, of course, it'll happen sooner. Otherwise, it will happen later. But it's my guess that what we're talking about is decades and not centuries. So that would be my answer about the time frame. The other question is, what can generations now do if you believe that you don't have a chance to immediately take advantage of technologies that could move you to other substrates? So if you're thinking of this as a life extension technique, but you're worried about that, well, personally, I'm not convinced that our generation would miss out. That's just a personal conviction. But if worst comes to worst, I think that we can consider brain preservation as a way to at least have a chance at future data extraction and whole brain emulation. So this is something that I think I should put out there. Julio, did you have any more questions for me?
No, I think Tudor had another question. We started more or less at the same time.
Okay, thank you. Randall, well, one of the questions is, what do you see as the tipping point event, as it were? You know, you have Werner Vinge and Kurzweil pushing the singularity as an important threshold, and it's in people's minds. Aubrey de Grey's pushing a Methuselarity, where he's basically saying for every year that you can add more than 1 year to life, you're going to have escape velocity for longevity. And here we can maybe talk about a scanality, or an event where resources, funding, researchers start moving into the field, and maybe that's a memory prosthesis, or is it finding the right level of resolution, or just doing a full scan? What do you think is that one critical event to get the real kind of resources that something like this requires, that we can all sort of strive for?
What's the tipping point there? I think that, yeah. Okay, one tipping point that I can imagine is in fact this proof of concept. If you can show that what we're talking about is possible at any scale, at any acceptable scale, let's say a very small brain or a piece of a brain, if you can show that, then at first maybe this will just incite interest in neuroprosthetics. But very quickly thereafter you get to the point where we're talking about, oh, why not do prosthetics for this part and that part and that part and that part, and it just kind of snowballs. So I think proofs of concept are very important, and what Ted Berger's doing is actually quite important as well there. Yeah, there may be other tipping points as well, but this is one that just comes to mind right away.
And Ted Berger has not achieved that yet. He hasn't done a proof of concept for a small piece.
I guess not. Not yet, no. Yeah, that's a good question, actually. We should try to figure out why is that not a much harder topic than it is right now, given what he's doing. Yeah.
Actually, we don't know Ted Berger's current state because very few people are able to get in touch with him, so we don't know exactly what he is doing. I myself tried to get in touch with him last month, and which was unique that he didn't respond because he usually is open to communications. But I had a question for you. On your website, you say that carbon copies takes a realistic route to substrate independent minds. And my question for you is, of course, in the chat, it was responded that by realistic you mean an actual process rather than a specific type of set of hyperboles which talk about uploading and whole brain emulation, etc. But my question for you is, what is your methodology or your heuristics or your strategy for developing a realistic route? What are those specific routes?
Well, the specific routes are really beyond. I can't really describe what they are at the moment because we haven't progressed that far. We have a number of different technologies that are interesting. We have a number of big fundamental questions. But really, it's not a very different approach than what you would take in any scientific approach, is that you try to analyze what are your problems, what are the problems that we face, then what are the possible solutions for those problems, and then you try to map out a way to analyze a number of different solutions and see how well they work, and then carry on that way. So really, our organization is not meant to advance one particular project. It's not meant to say, okay, we love this approach, so we're going to jump on that bandwagon. It's exactly the opposite. What our organization is trying to do is to step back and always look at the objective and always look at what is the verifiable evidence coming out of a specific project. What can we use from that? And then put it together.
So basically, what you're saying is your realistic route is the strategy is just the same type of business strategy or futurology strategy that we all employ in assessing the environment with the specific terminologies of who our stakeholders are, what the environment is, what the situation and different possible strategic routes to take, and looking at what's available. So you don't have a specific viable strategy at hand for producing the carbon copies.
No, we don't have a specific strategy at this moment. No, what we have is an approach that should identify the viable strategy. I think it's too early to jump ahead and say, I think it's too early for anyone to jump ahead and say we have a viable approach, because we simply haven't asked many of the fundamental questions. That's one of the first things that needs to be done. So it's a matter of producing literature. It's a matter of producing some networks between people who understand different aspects of the problem. And it's a matter of putting together different projects, some that exist in the mainstream, some that...
So what you're doing is you're developing a knowledge base library of information from which to draw.
It's a knowledge base, it's a network, a knowledge base, and support. So support to people who are doing things that are interesting to us, that help us along, yeah.
Thank you, that answers the question. So it's a knowledge base. Thank you so much.
I think I'm going to ask a question on behalf of Amara. And I know that the answer to the first one is going to be interesting. What do you think, and could you discuss the hammer of Penrose model? That's what Amara is asking. And as soon well, I know that there are some people in the audience who have some quite strong feelings about this, but just answer the first, and then I'm going to go on with the second.
Okay, can I discuss the hammer of Penrose model, besides the fact that it seems to be constructed merely as a way to express, oh yes, this is an exotic piece of hardware because it does exotic things that I can't understand, therefore it must involve something else that's exotic and difficult for me to understand. Hence, quantum computing must be necessary, quantum mechanics must be essential for what's going on in the brain. That seems to be the main connection that's being made there, with no real evidence for any particular function being carried out by the brain that requires this quantum mechanical leap there. So far, everything that's come out of neuroscience, everything about specific functions that the brain carries out and behavior, all of it seems to point to very classical mechanisms. There's a really question in this case of applying Occam's razor. I don't have any proof that quantum mechanics doesn't play some significant role for function X, let's just say, of what the brain is doing, but it's clear that we can also explain this in much more classical terms. So applying Occam's razor, I don't see where Hameroff-Penrose really comes into the equation. I don't have an opinion about it beyond that. I think there was another question.
Thank you very much. And now the person who was asking the question is in the public text chat, so she will see whether the answer was clear and whether she agreed. Her second question was, could you comment on organic versus inorganic substrates?
You mean as a target substrate, organic versus inorganic?
I think so.
What kind of a comment would be required besides stating that both organic and inorganic compounds can be used to do computations, therefore both could be target substrates for carrying out the computations necessary to support a mind? There's no real, I mean, you don't have to favor one over the other, except if you have specific goals in mind. Inorganic may be better for some environments or some types of computation, but yeah.
It's kind of funny, though, that that question was asked. Just excuse me just a moment. It's kind of funny that that question was asked because it brings to the table issues about, you know, when we purchase certain foods that they're organic, and it would be interesting with this whole, you know, copying brain or brain emulation or uploading, etc., whatever terminology we use, that some people may vie for. Natural, and see how natural, the concept of natural fits in there, as well as organic. I want an organic brain upload.
Yeah.
But I have a question for you regarding upload and downloading. You know, sometimes the terms are used intermittently, and I was reading something recently about the origin of uploading was downloading, and how there's some squabble in the academic world, especially in areas of cognitive and neuroscience that engage HCI, and probably stemming maybe from perhaps from Van Forrester and the VCL in Illinois. But what is your view on the interchangeability of the terms uploading and downloading?
My view is that I like to stay away from both of them because they're confusing. This is
Yes.
the thing. The whole term
Yes.
uploading has been a very confusing term, not just in the sense of what it makes people think about, because they immediately get references to the matrix and stuff like that, but also because, yeah, as you say, uploading, downloading, which direction is it really? What are we talking about? Why not just scrap these unclear terms and talk about clear things, like whole brain emulation as a technology, or substrate independent minds, transition.
Yeah, that's a good one.
Substrate, things like that. Let's not talk about uploading or downloading.
Excellent, thank you.
Oh, by the way, I have one more comment on the Ted Berger issue, because Natasha mentioned that it's hard to get information out of the Berger lab, but it so happens that in one of the projects that Carbon Copies is affiliated with is a project to emulate the activity of C. elegans. And the people involved with that include 3 SU students, and one of them is Rosa Chan, who works in Ted Berger's lab. So there is, in fact, a channel there by which we may be able to find out more detailed information about what actually is going on right now.
You know, it's so interesting that you mentioned that, because I'm a visiting scholar at 21st Century Medicine, and the very project that is secretive, what we're doing is, of course, with C. elegans, because they're the easiest to get, but we're going to be working with memory. And in working on this project, getting ready to go forward as soon as I finish writing my dissertation, is that the C. elegans can't work individually. They work in groups. So you can't train a C. elegans individually. So I have to buy a whole group of C. elegans. And I thought that was very interesting. Talk about a mind think.
as Peter Passaro would probably say, if he wants to pipe in, C. elegans, there are many reasons why C. elegans is an odd one to pick if you're going to try to show something like substrate independent minds or whole brain emulation. And it's not just what you're saying about their cooperative, their need to be cooperative to get anything done or to learn anything, but also just the way that their nervous system works. And I won't go into that right now in detail, but there are some reasons why that may not be the perfect proof of concept.
Randall, I'm exchanging message with Amara, who apparently has some questions for you in the private text chat. She sent some IM directly to you. So in order to see that, you just right-click on the first name that you see on the list on the right.
It's open here. It's open here, but if this is the same Amara, the first question was the one about the computations, Kurzweil, and the second one was, what is your working assessment of how deep? Okay, I'll have to ask a little bit about that, but anyway, and then there was the Hameroff-Penrose, which we've just discussed, and organic versus inorganic. So the one question remaining would be, what is your working assessment of how deep? And can I just ask for a slight bit of clarification? What do you mean by how deep? What specifically how deep? Do you mean in terms of the resolution we need, or do you mean something else? Amara?
I think that microphone is not working. Also the first time. Okay, right. I'm also, yeah, here she's on the public chat.
Okay, let's see. What level of structure? Okay, so that's basically the resolution question. Is at what resolution would we need to investigate the structure and then maybe the function of what's going on? I have my personal view, and this is one shared by a number of people I've talked to about this recently, is that when it comes to describing the transition of the state of a neural system like the brain from one moment to the next, it may be sufficient to describe what's going on in terms of spike times. So if we know the spike timing for each neuron, because the neuron basically just sits there in its environment, and all it knows about what's going on in the world is when it receives enough activity that it feels the urge to spike, and then it does. So that's really the world of the neuron, and this seems to be a fundamental level. It involves using the time dimension, because we need to know when it happens, and the resolution in space is, okay, where is this neuron, what is it connected to, which other neurons is it connected to. I do have the feeling, though, that when we talk about the update of those transition functions, when we talk about how does it all change, that it may be necessary to look at higher resolutions, so to see what's happening at the synaptic sites, for instance. I see discussion about nanobots and sandwiches. Okay, that's not exactly a question, but sounds tasty. Is there anything else? Could I comment on the Yale study just announced regarding the effect of electrical fields? I'm afraid I haven't seen the Yale study, so I can't really comment on it. It's a good question somewhere, but what is the good question? Suzanne, which one are you referring to?
Hi, sorry, I just had to plug the mic in. I was referring to Amara's question about the Yale thing. So I don't actually know what this study is. I haven't seen it, but I'm imagining it's something to do with electrical and magnetic effects between neurons, may be important. And that would be something that would be very difficult to model.
But we're talking about electrical and magnetic effects in a solution that is highly conductive. So this is not quite the same thing as being subject to electric fields in, say, air or something like that. I don't really know what this will cause. But my guess is that, you have a pretty good conductance barrier. I don't think that electric fields will work quite the way I would expect. I don't know. I really don't have a particular comment about it, since I don't know the study. But I'm sure that the one thing I can say is that we can't rule out that there are things beyond spikes that are affecting what neurons are doing. Like we can't rule out that there are other diffuse chemicals that are affecting neurons, or that there are electromagnetic fields that are affecting. In fact, TMS.
Well, could I make a quick point about that, actually?
Yeah, sure.
Is that people talk about when they move down from the spiking neural net models, and they say, well, maybe you need to go further down in your level of simulation. People seem to jump straight to these quantum effects without worrying about actual classical EM effects that may also be just as hard to simulate as quantum effects. So.
And that's a good comment. Yes, why jump straight to quantum when you have so many other levels in between? Yeah.
Yeah, I mean, if you've ever tried to do a 3D electromagnetic simulation of between two conductive surfaces, it's actually quite intensive computationally.
For everyone, I have just the web page with URL given by Amara on the screen here on the left. And there is article, study shows electrical field influence brain activity. Yeah, I had seen it before, as a matter of fact. So this is what she's referring to, Randall.
Okay. Yeah, I'm not going to go read it right now, but perhaps someone can quickly summarize what was the finding. Maybe Amara could.
Yeah, I'm going to do that. I think Amara's microphone is not working yet very well. It's her first time in Teleplace. So what it says is that electrical field can influence the activity of neurons. Yale scientists report on July 15 issue of the journal Neuron. The researchers introduced slow oscillation signals into a brain tissue and found that the signal created a sort of feedback loop with changes in electrical field guiding neural activity, which in turn strengthened the electrical field. The finding helps explain why techniques that influence electrical fields, such as transcranial magnetic stimulation and deep brain stimulation, are effective for the treatment of various neurological disorders, including depression. The study also raises many questions about the possible effect of electrical field, such as power lines and cell phones, in which we immerse ourselves, saying David McCormick, some researcher and some important person at Yale. The ability of electric fields generated by the brain to influence its own activity appears to be particularly prominent during epileptic seizures. However, the study also demonstrates that the electrical fields also influence brain function during normal activities such as sleep. McCormick says the findings change the way in which we view brain function and may be of significant clinical value in controlling epilepsy, depression, and other neural dysfunctional states. Okay, now since this appeared on Kurzweil AI, I guess there is a link to the original. Here it is.
Is it a study that uses TMS? Is it a TMS study? Or was this about general electromagnetic fields that are in our environment? Was this about TMS?
She says no, not a TMS study.
Okay, that's interesting. Yeah, in that case, it's very hard to predict what the electromagnetic fields are if it's just the general environment, because there are, so many interfering electromagnetic fields, and exactly how much it can affect what's going on. I mean, this is a distributed network of neurons, many of which can fail to do exactly what they need to do at a specific time, and you still get the function done. So you've got a lot of robust activity there, and exactly how much a field can do that is unpredictable, changing in time from various different orientations. This is not a TMS study, so we're not giving it a strong field in one direction. It's not as if you're just making the pyramidal cells fire more strongly or less strongly based on the field and what it's inhibiting or exciting. If it's just environmental influence, it's really chaotic in a sense. So
I think there are a couple more comments in the text chat.
Yeah, actually that's going quite fast, and I'm having a hard time keeping up with it. I see Amara has a whole bunch of questions. Which one shall we pick? What is standing between us and actually going for a proof of concept? Everyone will tell you that, of course, funding is always something standing between us and doing anything, but aside from that, nothing. There's nothing standing between us and going for a proof of concept. And so, in fact, there are a number of people that we work with who are intending exactly that, to go forward and do a proof of concept. It's just that it takes time and it takes investment. So, those two things. And Suzanne says computational hardware as well. Yeah. Oh, wait, Amara says that it wasn't related to the environment. So what was it then? It's not a TMS study, and it's not related to the environment. Well, I guess I'll have to read the study, and I'll understand what it's about.
Randall, one clarification. You said that that carbon copies would be a foundation knowledge base, a network. Is that not just researchers, or is it also interested funding other foundations or individuals? Is it on two levels?
It's
both?
a broad network. It's basically just a needs-based network. We make the network as far as we need it to go. It's not just based on scientists, it's based on everything, really.
Okay.
Okay. Not sure if there are any more questions. Yeah, how do we get the funding? Yeah, good question, Eugene. We'll see.
Can I suggest that we do something like, robbing a bank, that kind of things? It seems like a quite expedient way of getting the funding we need. And, well, I hope some alternative ways of getting funding will materialize on the horizon before we take that option seriously.
Yeah, we'll have to discuss that in more detail. I don't think that we've really resolved the issue of how to go about the funding at this moment. The hypothalamus, yeah, that's a funny one. That looks to me like we might stop the recording sometime soon. No new questions coming up.
I would have a question.
Yes?
Would there be something, or is it possible that there might be something equivalent to epigenetics in neuro research?
Yeah, I really can't comment on that. I don't know. I'm sorry. Yeah, Eugene is pointing out that Limnia is reachable with today's hardware. Okay.
Hello, one question. Do you hear me?
Yes, I can barely hear you.
Okay, I try. Do you plan to make a sort of carbon copy from more easy animals like a fly or like a mice, mice, in the first time, or is this a project, a possible project?
Okay. Carbon Copies itself as an organization doesn't run a project, but we do affiliate with projects. And yes, there are proof of concept projects that would look at simple organisms, mostly invertebrates first, as a proof of concept for emulation. And this is then, for example, limnea, which is what Peter Passaro works in. I think that, yeah, a fruit fly or something like this, or something at some point, is already much more complex than limnea.
And the second question, you said that you were pretty sure that it was possible to have this copy on a non-organic substrate, but I know that there are controversies about this. So what makes you so sure, or why are other people not at all sure? Can you maybe summarize the arguments about this?
Well, what exactly is it that you're worried about in the first place? I mean, this inorganic versus organic question again. What makes organic processing special? Why would that be something that you couldn't do inorganically? I mean, what stops us from modeling anything and basically simulating what's going on a computer? As long as it's something that is Turing computable, something that, you know, that this is why I start with the Church-Turing thesis, as we assume that we have a machine that is doing all that hard work for us that creates our mind. And if it is in fact a machine, though a complex one, then you can implement a machine that carries out those functions in an inorganic substrate. So what exactly is it that you were worried about in terms of what an organic computation may be able to do that an inorganic cannot?
It's not that I worry, it's that I heard people saying that it was really impossible. And I try to understand why these scientists are saying that it's impossible, and why for you there is absolutely no problem.
Maybe they weren't referring to it being impossible to do on inorganic hardware. Perhaps they were really trying to say that we don't know enough about the organic components so that we can properly emulate their function in an inorganic substrate, that we just don't know how to make the right model yet, which is true. I mean, we don't know enough about these components so that we could make those statements clearly and say, yes, I can now go ahead and build you a model of this, this, and this neuron and put them together in a way so that we have a functioning limnea. I can't say that right now. So in that sense, they are correct. It is currently impossible to do. That doesn't mean it's impossible in principle, and it doesn't mean that there's anything about the inorganic substrate itself that would make it impossible. And if they do say that, then I would really like to know what they're trying to get at.
Okay, thank you for the answer.
Move to inorganic substrate, it would be hackable. That's a really good point. In fact, that's wonderful, isn't it? We would want it to be hackable, because that's exactly what we want. We want a substrate that we have access to, where we can do things like augment it to make it better. Right now, the fact that my brain is not hackable is something that really bothers me. So I think hackable is good. It's not just something dangerous.
So maybe the last question from me anyway. If you can carbon copy a human brain, so I suppose this human brain will work a lot faster. So do you think that it's a risk, or do you think that it's great? So you know about
okay, this goes back to that whole question. Why do you do this? Okay, first I'd like to say that probably the first implementation of a emulated brain will not be working faster than a real brain, but may in fact work a lot slower. For example, you saw the work that Eugene Etskiewicz did to run his simulation for 1 second, took him 50 days. So emulations in different hardware, as if you're emulating Windows on a PC, on a Mac. If you're emulating Windows on a Mac, often it'll slow down Windows because it doesn't run as fast on a different hardware. So the point where we actually get a faster emulation than the biological brain that's not necessarily immediate. This is something that comes after a while. But you're right, at some point you would. The hardware improves and eventually you get faster brains. This isn't a risk so much as it's a necessity, because we need to understand more about our environment. The pace of technology, the pace at which we have to understand what's going on around us is increasing rapidly, and we want to be able to understand more. And we want to be able to comprehend also what our machines are up to. Let's say if you have artificial general intelligence that is making use of such hardware, then you would want us to be able to make use of that as well. Basically, you just get a speed up. And that's something we've been seeing over the past 100 years, and we're going to see more of that in the future. I see a lot of discussion about hacking on the text chat now. How do you cause a buffer overrun on a dog? Okay.
I can get my own dog to participate in the discussion. She might have some strong ideas about these things.
Yeah, it's too bad that the text chat is not visible on the recording. It would be a nice, it will go well with everything else. Yeah, that's right. It would be easier to read like that. Yes, Suzanne. It's, oh yes, it can be exported. So we can at least keep the text chat, but we won't know exactly which part of the discussion it was related to. Sorry, could I comment on what? I'd like to hear thoughts on measuring and emulating radio frequency signals from the brain. I guess you could pick them up in any sense, just the same way you would any other radio signal. There's nothing exotic about radio signals emanating from the brain. And emulate them in the sense, well, you can take a radio signal and you can teach a computer to produce the same radio signal or the same signal shape. So there's nothing particularly bizarre about that. It's just that I'm not sure what you mean by radio signals from the brain, because what are they beyond some chaotic mess of radio frequencies that are emanating from billions of little computations that are producing all kinds of electrical activity, and this is all being pushed out through the skull, which is already attenuating the signal and causing further changes in the signal. So I'm not sure exactly what you mean by the radio frequency of the brain, besides a basic chaotic mishmash of radiation. ELF signals. Ah, okay. Well, what are these low frequency signals? What are they from the brain? I mean, I don't know about what the brain is producing in terms of ELF. Yeah, that's kind of what I was thinking. There's no particular information coming from that. If there were, this would be fantastic, because I mean that would be access to some information we could use to extract the data for our substrate independent mind transfer. But I really don't see that there's anything coming out of this. I mean, if you look at EEG, those are really only the really big general global signals are even comprehensible. The rest is all just noise. Eugene, yeah, I don't know. I haven't really decided what I'm going to talk about in San Francisco. This is way too long, of course, so I probably have to cut down to very specific things, and I probably want to aim more at the organizational stuff and maybe sort of introducing what's going on and maybe some talk about fundamental issues. Not so much all the other stuff, because other people will be talking about bits and pieces, so not quite this talk. Yeah, we should make it more compact. Yes, I agree. This was very long.
Yeah, given that many people have a kind of attention deficit these days, the ideal would be a 15-minute talk. But I really don't see how anyone could say all these things in 15 minutes. Unfortunately, there are things that have to take 1 hour.
Yeah, well, at least once in a while you need to go over the whole thing, just so that it's there as a reference, and then it's possible to make specific presentation on specific topics and questions, just like the questions that are coming up right now. We could address each one of them with an individual talk, basically, about just that. So, you're interested in the optogenetic detection of consciousness. Any comments? Oh, well, that's a big interest about 2 different topics. Optogenetics being a technique for addressing specific neurons in the brain, or specific types of neurons, for example, and consciousness being a completely different topic, really, about what is consciousness, first of all, and how could you possibly detect it, and besides just detecting when consciousness is present or absent. So this is, I mean, you could use optogenetic techniques to detect certain brain rhythms, and therefore you could use it as a way to maybe make a causal relationship between these brain rhythms and the state of consciousness that you're in. And in that sense, I see a connection there. Okay, I'm going to be happy to continue this conversation. I just need a 1-minute break, if that's all right with everyone. So I'm just going to turn off my camera for a second and my microphone. I'll be right back.